I'm presenting a report from a section of what I've been doing the last 3 years in my PhD. My PhD has involved an exploratory laboratory investigation of the influence, the hydromechanical influence that the growth of fungi would have in soils, specifically looking at sands. And today I'm going to be talking about the influence of this fungal growth on the erodibility of sands. Soil erosion is a very huge problem across the world right now, and this study in 2001 has been able to quantify that 130 billion metric tons of soil is translocated due to water-induced soil erosion across the world. And recently, in a symposium organized by the Food and Agricultural Organization, the deputy director in charge of natural resource conservation had said that in every 5 seconds, soil equivalent to the size of a football field is actually lost globally. And if this trend continues up to 2016 without much being done to stop it, about 90% of our lands would be degraded at that time. And if we see also that the formation of about 3 centimeters of soil takes close to 1,000 years, it also drives home the point that this is a very urgent problem. The annual costs of damages and prevention of erosion also continues to increase. This is just data from about 20 years ago, showing that $44 billion is spent on soil erosion and damages on-site and off-site and the different forms in the US alone, and in the UK up to 90 million pounds as of 2002. Currently, this would definitely be far more than that amount. And the effects of soil erosion cuts across geoinfrastructure, agriculture, and the environment. For instance, in embankment dams and issues of instability, it's usually triggered with surface erosion. Beginning of all of this is from surface erosion. In agriculture, the loss of the nutrient-rich topsoil means that we'll probably be having hunger in a very short time if nothing is done. Environmentally, the issue of land degradation comes into play, and the fact that we have siltation of water bodies and the issue of nutrient enrichment in downstream water bodies leading to the cascading ecological effects of algal blooms, loss of aquatic lives, fish kills, and general ecosystem imbalance. It is a very big problem. And the traditional approaches that have been used before now have been found to be competent and have done quite well so far, but the problem still persists. Some of the traditional approaches that have been used include the installation of drainage to take surface and subsurface water off, the use of geosynthetics and geomembranes for also filtration and reinforcements, and also chemical-based soil stabilization using chemicals like cement, lime, fly ash, xanthan gum, polyacrylamide, which are organic compounds as well. But the problem with these techniques, some of them are actually energy intensive, some are expensive to deploy to sites and for maintenance. They mostly require heavy machinery, they disturb the ground, and feed into environmental hazards in different forms. For some of the methods leave harmful chemicals in the soil, like cement, for instance. And of course, the issue of low carbon, which cement is a very huge culprit for civil engineers at the moment. So with the clamor for low carbon technologies because of climate change and global warming, engineers are beginning to transition to use of materials that involve low carbon, materials that are technically effective still, while being low carbon, socioeconomically viable, eco-friendly, and resilient to climate change over time. And in this, we begin to look at natural engineering, looking at bio-based methods for control of erosion. And what has been done very well currently and over time is the use of vegetation in erosion control. And this is because the vegetation, which you've heard of a lot about in the last session, brings into the soil some hydromechanical effects in terms of the shoots and the roots, help to keep soil reinforced, especially the shallow roots of soils which have been associated with higher tensile strength in the soils. And furthermore, people have begun to look at using some species of fungi, they are buscular mycorrhizal fungi, along with the roots of plants to help further create aggregates to help the plants grow better, create a good environment for the plants, while the hyphae of the mycelium, which I'll talk about very shortly, also contributes in its own way to helping to keep the soils together and reducing the loss of soil. There are also studies, few of them, that have looked at the use of bacterial biofilms and microbially derived exudates, such as the exopolymeric substances, which also have some binding effects on soil particles, thereby reducing the loss of these particles. And more currently and popular, we have the microbially induced calcite precipitation, which is a type of biomineralization that is being used now to basically turn things like sand into sandstone, getting soil really held together and reducing what happens with erosion. In all of these, there's been very few studies that have looked at what could happen with some other species of microbes in the soil, which I'll talk about, which is the fungi. But this picture here is a biological soil cross, and they've been found in arid zones to reduce the erosion of the soils. This soil cross contains bacteria, algae, and fungi in different proportions. And the effects that fungi alone has seem to be more amplified from what studies are beginning to find out. Because fungi on Earth is actually massive, and in soil is actually also massive. 25% of the total biomass on Earth is fungi, and in the soil alone, up to 75% of the total microbial biomass is contributed by fungi. It's like breaking news, so many jaws open. And yeah, I'll shock you further, the world's largest organism, and the oldest. This was confirmed just 3 weeks ago, about a month ago, by Nature investigation, that a species of fungi is actually the oldest organism on Earth, about 1 billion years old. And there are over 10 million species of fungi. And you may have heard that fungus is quite hazardous and dangerous to man and to vegetation and all that, but of the 99,000 species we know, less than 10% are pathogenic or parasitic to man or to animals. And this is why we're beginning to come into the geomechanics, geotechnical engineering community and say, okay, if this is in soil this much, there is some potential in using it for the various ground improvement techniques that you do, and especially in soil erosion, which I'm talking about today. Where this is all coming from is based on the hypothesis from soil science, who have looked at arbuscular mycorrhizal fungi, which grows with plants, which is what is popularly studied in terms of what it do to soil in aggregating soil particles. But I will be talking about the filamentous species of fungi, which grow by formation of hyphae. Hyphae are just thin tubular structures which tend to be between 1 to 35 microns in diameter and can grow from several microns to several meters in the soil. And when they grow together, they form these three-dimensional networks called mycelium within the soil. This is just an image of a microscopic image of hyphal networks holding soil particles together. So the hypothesis upon which we're building this whole studies is from this description of what hyphal networks do in terms of aggregating soil particles, as described by Rillig and Mommy in 2006. And the mechanisms involve physical, biochemical, and biological mechanisms. Physically, the hyphal networks tend to immerse soil particles, holding them together, and also affecting how water retention properties of the soil. And biochemically, the exudates, as the fungal hyphae grows, they release certain exudates that are gummy, so they sort of help to bind soil particles together. And they also alter the wettability of the soils. Some of them make soils hydrophobic, others make it hydrophilic, or even alternating the hydrophobic properties. And biologically, where the fungal species are growing tend to affect other biological community, other microbial community within the soil in terms of bacteria and even plants, depending on the species. This has been described using arbuscular mycorrhizal fungi that grows with plants. But we are looking at a saprophytic species of fungus, which I'll describe very shortly, which actually do not need plant hosts to grow. They actually feed on dead organic matter in the soil and may not be as restricted as the arbuscular mycorrhizal fungi. there's not been so much study in applying fungi to erosion in the soil. But the few we've been able to filter out, we see that most of the studies, like I said, have been using arbuscular mycorrhizal fungi. And this is indeed difficult to pick out what the contribution of the fungus is from what the contribution of the plant is. There's been a lot of debate going on with that. And also these species require plant hosts. And plant hosts may be restricted in terms of where they grow and the requirements and things like that. There's been also another study that's seen that the influence that fungal hyphae that are non-arbuscular mycorrhiza would have in soil may not be too far from what you get with the ones that are arbuscular mycorrhizal fungi. So we have also used that to look at, this study has also tried to look at what saprotrophic species, the ones which are not restricted by plants, tend to also bring into the soil. And this is probably the only study that we found that looked at erosion using fungi, but they investigated wind-induced soil erosion, and this was done just by interpreting the abrasive resistance of soils. In the first study, they've been looking at majorly using aggregation to interpret how it helps to stabilize soils against erosion. And this study has used native fungal species, that means existing species in different natural soils, to see what their contributions would be. It's also not very clear how the fungal contributions and mechanisms have been. We are thinking about getting a species of fungi, engineering its growth, and seeing if it's going to be brought into the site or developed from the lab and taken to site and all of that at the beginning stages of it. So what I'm presenting today is just filling the gap of study in the area of looking at the influence of a single species of fungus, which is Pleurotus striatus, on water-induced soil erosion. Pleurotus striatus is the second most consumed mushroom, most cultivated mushroom across the world right now. It is very, very safe to man and to the environment. We're not concerned about this mushroom actually, we're looking at the hyphae, the part that grows within the soil. And we're also in this study for the first time using a sort of more acceptable erosion testing method to the engineering community. So the objectives I'm going to be addressing basically how we have used the jet erosion testing apparatus, which is the equipment we use to determine the erodibility characteristics of fungal treated and untreated soils using the Pleurotus striatus fungal species, to also determine the effect of inoculation methods and effect of growth duration on the erodibility. We have just concentrated on sands for now as a startup. This is a very sort of new area, and it's easier to see what is happening with sandy soils than when we look into other soils that might have more complicated factors, for instance. So we prepare our space. We developed a method, sort of protocol for having the fungi inoculated into the soil. We have the fungal spawn from which we remove a fixed mass and add into deionized water in this conical flask and stir and shake for about 30 minutes. So the spawn actually contains grains of millet colonized by the fungal species, Pleurotus striatus, and this is just shaken to extract or sort of harvest the spores from the spawn, and then we filter that through to get a spore suspension. The spore suspension is then added to the fine sand and lignocell. Lignocell is just a source of food for the fungi, organic matter, fibers. And then for the control, we use water instead of the spore suspension. And this is produced into the specimen, which is in the mold. The mold is 100 millimeters in height by 100 millimeters diameter. This mold is used because it fits into the apparatus, which I would describe very shortly. We produced specimens in these forms just because we want to investigate the inoculation methods, looking at mimicking certain field applications possible. This, for instance, represents untreated. The darker brown just represents the spore suspension. So for fully treated, we have all through the soil profile, soil mixed with the spore suspension. For the half treated, we just have the top half of the specimen mixed with the spore suspension. And for this surface treated, we sprayed through on the surface, making kind of hydroseeding or something like that on the surface of the specimen, while the other parts have just water. So for the first objective, which is to determine the erodibility characteristics, we prepared untreated specimens in triplicate and fully treated specimens, that's this one and this one. in triplicate, just to investigate what's happening with changes in erodibility. And to look at the effect of inoculation methods and growth duration, we now had the fully and the half and the surface treated specimens prepared across 1 to 9 weeks, 1, 3, 6, and 9 weeks for all of the specimens. So after preparing the specimens, they're incubated in the incubator at 25 degrees in the dark so that we don't have mushroom growing, just for the hyphae to grow. And after the specific time allotted to each of them, they're brought out and placed in the jet apparatus here. This is the jet apparatus we developed here at the University of Strathclyde. Well, based on the standard ASTM jet apparatus, jet erosion test apparatus. It has this jet tube, and the specimen is placed somewhere around here. We have a deflector which helps. The jet tube is filled with water at different hydraulic head, and an impinging jet comes down on the surface of the specimen, which is placed, the specimen is placed in a submerged tank filled with water. And so the theory of the diffusion of an impinging jet is what is used to describe it. I'll talk about that very shortly, quickly. And then, well, I'll just go quickly now. This is just a schematic showing the stress distribution and how the diffuse jet theory is applied to it. So basically, erosion is described using the excess shear law, the popularly called erosion law, relating the erosion rate to the erodibility characteristics. That's the critical shear stress and the erodibility coefficient. And this is the typical plot that is used to describe different levels of erosion that you have for different soils. But because this law requires a kind of equilibrium scour depth to be attained during the erosion process, several equations and methods have been developed over time to be able to determine ranges of erodibility of soils just by plotting the erodibility coefficient against the critical shear stress. I will just go very quickly now because of time. And then during the process, all you get from this whole analysis is the scour depth during the erosion test. You get the scour depth and put it into the equations to get the erodibility coefficient and the critical shear stress. But we thought that for the kind of specimen we're working on, there may be a need to have an idea of the volumes of soil that is eroded. And so we take our specimens also during the procedure into the laser scanner to get the surface profile, which helps us to get the volumetric difference before and after the tests. This was meant to be a video to show how a plot of the scour depth obtained at different hydraulic heads changed in the jet tube against the time of the tests. And this is for one untreated specimen. We begin to see how the erosion, the scour, actually begins immediately. The test starts. Within a few minutes, sort of an equilibrium scour depth is achieved. And when the hydraulic head is increased in the jet tube, the scour depth again jumps and then quickly attains an equilibrium erosion scour depth, and then again up to 250 millimeters. I stopped at 250 millimeters because we already had almost 70 out of 100 millimeter depth in the soil. When we tried at 500 millimeters, just within 2 seconds, the entire soil had eroded. And this is just images of the soil before, and at, like I said, hydraulic head of 100 millimeters, that's here. After 120 minutes, we had this level of scour depth, and at 250 millimeters, and at 500, just 2 minutes after, we have everything eroded. For the three others, the replicates, the difference is not really quite much, significantly when we tested with some simple statistics. And I've brought in here the blue points representing the treated specimens. And we begin to see that compared to the untreated specimen, there's a delay, there's a lag in scour being obtained. And even when scour did start occurring, it was a bit slower, not as fast as the untreated one in getting to the equilibrium depth. It scoured gradually before getting to the maximum scour depth we obtained. And then generally you see this cluster of the treated specimens showing that far less scour depths were obtained for the treated specimens compared to the untreated specimen. Again, looking at the profile of the pattern of erosion we have, we see that for the treated specimens compared to that, we tend to just have a scour hole caused by the impinging jet right there in the middle, compared to here where we have large volumes being eroded out of the mold, showing that there's some level of binding due to the fungus within the specimens. And this even becomes clearer with the laser scanning images which we took and the volumes which we removed. I'll show you that very shortly. You'll see that larger volumes are actually eroded from the untreated specimens, and then you just have these scar depth volumes occurring in the treated specimen. This is a plot of the percentage volume eroded which we extracted from the laser scanning against the hydraulic heads, changing the different hydraulic heads. And again, clearly, this shows that all of the treated specimens were far less volumes of the treated specimens were eroded compared to the untreated specimen. For instance, at hydraulic head of 250, where almost 50% of the untreated specimens are eroded, just about 5% of the treated specimen had gone. And similarly, at hydraulic head of 500, where the untreated specimen, actually none was existing, we had just still less than 5% of the treated specimen being eroded. Also, in comparing the erodibility characteristics, I'll just use this plot, which is the erosion chart of the erodibility coefficient against the critical shear stress. Basically, the higher the critical shear stress, the less erodible the soil is. The higher the erodibility coefficient, the more the rate of erosion is occurring. We see the cluster of the untreated soils in red points here at areas where you have higher erodibility and much less critical shear stress compared to the treated ones. And you also note that for this analysis, it's just the scar depth that is being used, so probably it's not showing very significant difference, but if you look at the volumes, which we saw just now, you begin to see that. But if we put these bands of erosion classification based on literature upon this data, we see that the untreated soils fall into the very erodible range, and the treated soils have been able to get into the moderately erodible range due to the fungal treatment with lower KD and higher TC. This is just a summary of laser scans for the control, the fully treated, half treated, and surface treated, just to show what's happening with the inoculation methods at hydraulic head of 250 and 500. We see that it's not quite significant difference between the different treatment methods, but then the surface treated seem to erode even more compared to the fully treated and the half treated specimens. And bringing the scar depth now into the plot of the erodibility chart, we again see the clusters of the treated and the untreated, but in this case, 3-week fully treated, half treated, and surface treated with those signs. These are the surface treated specimens falling between very erodible and erodible mostly, and the fully treated up there and the half treated somewhere, showing that they're more resistant, well, marginally, not very significantly. But then it's clear to see that surface treated specimens tend to perform least in terms of erosion resistance compared to the half treated specimen. And then looking at the influence of inoculation method and growth duration on erodibility. Here I'm comparing the maximum scar depths at the different hydraulic heads with the percentage volume of soils eroded. While the scar depths tend to show that, still the treated specimens are less erodible compared to the untreated specimen, we see that the 1-week full treated specimens tend to behave like, or even worse than, the untreated specimens here. But looking at the volumes eroded, we see that that's not very significant misbehavior, if you would say, in terms of the 1-week fully treated specimens. Again, if we look at the half-treated specimens alone, in this case we're looking at the full treated specimens. If we look at the half-treated specimens, it's similar to what we just saw. Again, the 1-week treated specimens seem not to behave well from what we see in the maximum scar depth. But in the volumes eroded, it's still taking the same trend. But still, the treated specimens still tend to behave even better. And if we finally look at the surface treated specimens, it becomes clearer that surface treatment tend to be the issue here, where we have the surface treated 1-week specimens all behaving poorly compared to the untreated specimens. And then we can conclude that 1 week of treatment is definitely not sufficient to get a good contribution to erodibility, and that the highest erodibility obtained is for specimens with 1 week of treatment and surface-treated methods. Okay, I would soon wrap up. So for the 1 week treated specimens, I'm just trying to zoom in on the laser scans to show why this is so and what we see here. These are fully treated, half treated, and surface treated. We see, for instance, hydraulic head of 100. This is massive amounts of volumes lost from the surface treated specimens, and also at hydraulic head of 250. This, we think, is probably due to less hyphal growth on the surface for 1 week, because the mechanism of hyphal growth and enmeshment of particles need time. And 1 week is probably not enough to grow through the profile of the specimen. And also, in the 1 week treated specimen, we saw that the aggregates the soil was being detached in forms of aggregates, compared to in forms of particles, which you would see even in untreated specimen. Probably this is why the surface treated specimen even behave poorly than the untreated specimen. And if we place all of this together on the erodibility chart, again, we see that in the very erodible band is shared between the untreated specimen and most of the 1 week surface treated specimens, and some of the 1 week fully treated specimens. We see all the 1 week treated specimens falling into that band, and most of the others are just here where you can't really pick out significant difference between the growth at that point. So meaning basically that after 3 weeks, or after 3 weeks of treatment, we see no significant difference in the erosion, the erodibility effects that the fungal growth has in the soil. For the mechanisms contributing to this, like I said, the hyphal enmeshment of particles, the exudates that come in from the fungi tend to bind the particles together and hold it together. And there is also the issue of water repellency, which I studied in one of my studies as well, where we've seen that this species of fungi contributes to water repellence in the soil. And since it's that way, we actually even tested another infiltration test where to get water through from the top to the bottom of a 30-centimeter soil column took almost 6 days with treatment with fungi. Then we begin to infer that the water repellency may have resulted in incomplete saturation during the process of the test when it was submerged, and then there will be less infiltration and maybe higher suction in the treated soil compared to the untreated soil, and therefore helping to bind the particles together. Also, one significant contribution we have from this study is that typically soils that are water repellent, as induced now by this fungi, always lead to higher erodibility. They're usually loose and you have more erosion. But we have been able to show that sands that are water repellent by the growth of the fungi also lower the erodibility of the soil significantly. So in conclusion, fungal treatment significantly increases the critical shear stress and reduces the erodibility coefficient, meaning that it improves the erodibility of soils, especially at 3 weeks of growth or more. And the growth duration of 1 week is not enough. Growth duration is significant. It's a biological system. And then we couldn't find so much significant difference in terms of the treatment types at 3 weeks or after 3 weeks. This is the beginning of a very big sort of study that could look at deploying fungi to the soil for erosion control and generally for soil improvement. So there are still lots of questions. For instance, we worked with sand, and you want to look at what will happen with natural soils if you're not growing the sand from the lab to move to deploy to the site. We've also looked at sterile sand. We've isolated the conditions under controlled environments in the laboratory. So there'll be a lot of things to do, a wide spectrum of tests and trials to go on before we deploy to field and then see things that happen there. But these tend to show that there is a very good potential in this technique for erosion control and soil improvement in general. I'd like to thank all of these people who have helped in different ways during the tests. Thank you very much.